Welcome to the last and the fourth part of gram-positive bacilli. So in this part, we will be discussing over the anaerobic gram-positive bacilli or the anaerobic gram-positive bacteria. So in this list, we had come across Bifidobacterium, Eubacterium, Propionibacterium, Lactobacillus, Actinomycetes, and Mobilincus. So briefly, let's see what are the diseases caused by them. So Bifidobacterium is mostly non-pathogenic. The Eubacterium may sometimes cause periodontitis. Propionibacterium, again, the pathogenicity is uncertain. Lactobacillus can be encountered in supragingival plaques. Actinomyces causes cervicofacial actinomycosis, and Mobilincus causes bacterial vaginosis along with Gardnerella. When infection with anaerobes is there, there are certain clues which indicate anaerobic infections. These infections are in continuity to the mucosal surfaces. Infections with tissue necrosis and abscess formation is there. There is a lot of tissue necrosis and abscess formation. The wound has a putrid odor. There is a lot of gas collection in the tissues, as we had seen in the discussion of Clostridium perfringens causing gas gangrene. Anaerobic infections are usually polymicrobial in flora. That means at the same time, multiple organisms can be uh, uh, isolated, or multiple organisms can be related with the wound. There is failure to grow in the laboratory under aerobic conditions. Routinely, we cultivate bacteria under aerobic conditions. So because these are anaerobes, these will not grow under aerobic conditions. So that indicates that maybe an anaerobe is present in the specimen. There is failure to respond to antibiotics that do not have a significant anaerobic activity. Then these organisms are seen under the microscope, but they fail to grow in routine culture because routinely we opt for aerobic culture. So in order to grow anaerobes, we need to have a high level of suspicion for anaerobes in the specimen. And sometimes these wounds are associated with special features like sulfur granules. These granules are present in uh, uh, actinomyces infections. Coming to the laboratory diagnosis with these anaerobic gram-positive bacilli, we can go on for microscopy. We can collect the specimens and we can inoculate them in Robertson's cooked meat broth, and then we can make a primary or the direct smear. Look for the presence of the bacteria, look for the presence of the gram-positive bacilli with different characteristic appearances, like we had seen in case of Clostridium perfringens, which is a gram-positive stout bacilli with absence of spore. Then we have seen in the case of Clostridium tetani, which is a gram-positive bacilli with a terminal and round or circular spherical spore. So in these cases, in these anaerobic gram-positive bacilli, these are non-sporing. We did not discuss it in the beginning, but these are non-sporing anaerobic gram-positive bacilli. So in these cases, we are bound to see gram-positive bacilli without spores on gram staining. Then we can move on for culture. Culture can be done on blood agar with added kanamycin or vancomycin. We can also culture them on thioglycolate broth or Robertson's cooked meat broth. Culture is done under excuse me. Culture is done under anaerobiosis in a Macintosh wheels jar. So we have a picture of a Macintosh wheels jar, and the plates are incubated at 37 degrees Celsius at least for 2 days, and thereafter completion of the incubation period, the plates are observed for growth. Any growth, if present, is identified. Now there are different biochemical tests, a range of different biochemical tests for different bacteria, which for different bacilli which are suspected. So we can perform the biochemical test according to the suspicion of the bacteria. That depends on the clinical symptoms too. And apart from this, we can also perform the gas liquid chromatography, which detects the metabolic end products, the specific metabolic end products which are produced by certain bacteria. And finally, for treatment, we can opt for metronidazole, penicillin, and clindamycin. And that's all for a very, very brief discussion on non-sporing anaerobic gram-positive bacilli. So with this, we end with all the four parts of gram-positive bacilli, and we complete the entire discussion of the gram-positive bacilli. Thank you.